Hello everyone. I hope everyone is safe and sound in this ongoing crisis. I request everyone to stay home and not venture out unless it is required to stop the spread of this infection. Now, being a master student in biomedical sciences, it is very important for me to give you a glimpse about this virus and how its structure is so important for causing infections. Now, here you can see a diagram depicting the coronavirus, which is more like in a spherical or circular in nature, and you can see that there are spike like structures present all around the surface. Now these spike proteins are very essential. These spike proteins are used by the virus to gain entry into a host cell. Now you must have heard about the angiotensin converting enzyme 2 or ACE2, which is present in host cells like in cells of the lungs, heart, and so on. Now these spike proteins lock onto these receptors or these enzymes, and this helps the host cells to open up. Once the host cells open up, it can go inside and release its genetic material, which is this one that I have shown. So these viruses, once they gain entry into a host cell, and then it can direct replication of its genetic material. Once the genetic material is replicated, it also produces virus-like particles. All these particles and the genetic material are assembled and new virus particles are formed. Once these virus particles are formed, these are released outside the cell, and now the new virus particles can infect new cells and get released into the environment. Now these spike proteins are very crucial for the virus to gain entry. If there was no such protein, or if the proteins are damaged, it will not be able to go inside the cell and infect, and thereby making the virus non-transmittable or non-lethal. Now these spike proteins can be blocked either by blocking the enzyme where it locks into, or you can destroy these spike proteins, which would be a bit difficult, but if you can block these spike proteins through some substrates which mimic the receptors that are present in the host cell. Now you have to come up with a substrate which will be similar to the structure that is present in the host cell. Once these structures bind to the spike proteins, the virus will not be able to infect the host cell. Let me also explain you how washing our hands and using hand sanitizer based on alcohol is so useful in destroying this virus. Now, everywhere in media and all throughout the world, focus is being given on washing of hands. Not only this washing, and they have also focused on a particular time limit, at least for 20 to 30 seconds. Now, how is that important? Now imagine you apply soap and you're rubbing your hands. While you rub your hands, you have to rub it fully all throughout, because this virus sticks to surfaces. Now these surfaces, the surfaces of the virus have to be dismantled. If you keep on rubbing for a long time, the viral surface integrity is completely lost. When the virus surface or the membrane integrity is completely lost, the structure is no more useful for the virus, and it will come out when you wash off your hands. This is why it is being recommended to wash your hands with soap and water for at least 20 to 30 seconds. Even hand sanitizers, particularly ones that contain about 70 to 80% of alcohol, is very important. Alcohol also manages to dismantle or dislodge the same membrane. What I refer to as dislodging the cell membrane is this structure is fully dismantled, or the structure is no more like this. Once this structure breaks or the spike proteins are released, all are like debris, and which can come out when you wash off your hands with water. And it is also important to completely dry off your hands, because even this virus can stick to surfaces which are a bit wet. I hope this helped you in understanding why is it so important and why is it being stressed right now to wash your hands regularly and sanitize it with alcohol-based hand sanitizers. I hope you liked it, and thank you for listening to this informational and interesting video.